the view with
Yep.
the whole deck on
Yep.
the side?
Just one
Okay.
slide. It's perfect.
Great, thanks. Okay, so thanks a lot for having me today. I'm Matt Sharkey, biologist at ASPR. I introduced myself before. I want to say that I'm particularly happy to be here because when I was a much younger man with many fewer gray hairs, I worked for Gene at Battelle on these issues, and that was one of the most formative jobs I've had, and I took that experience through several subsequent jobs, and am lucky enough to have ended up working on the federal guidance that suggests that these types of screening protocols are used for providers of synthetic nucleic acids. I'm going to give an overview of the 2010 guidance from HHS, and then I'm going to give a forward-looking summary of changes that should be expected in the guidance in the coming year, and the issues that have come up given the evolving technology landscape. So kind of the why. Why does the federal government want providers of synthetic DNA at this point to screen for sequences of concern? Probably you guys don't need this background, but prior to the advances in synthetic biology that occurred over the past 20 years, 30 years, really, there was access control inherent in the availability of entire pathogens, especially entire viruses. So we didn't have to worry prior to 2001, when the first synthetic poliovirus was produced in HeLa cells, that if we had access control for a small RNA virus, that people who were unauthorized to obtain those viruses could get their hands on them after the synthetic poliovirus genome work was published in 91, and then synthetic poliovirus itself was produced in 2001. There was more of a concern that the Select Agent Program and the Department of Commerce's Commerce Control List would not be able to prevent unauthorized people from accessing those. So I'll talk subsequently about advances in 2017 that have led to a revision of the guidance. But oh, I'm sorry, I'm advancing the wrong set of slides. Okay, so in 2010, in order to reduce the risk that unauthorized individuals or individuals with malicious intent could obtain regulated agents, those regulated agents being listed by the Federal Select Agent Program or Department of Commerce through the Commerce Control List, through the use of commercial DNA synthesis, the Department of Health and Human Services issued the screening framework guidance for providers of synthetic double-stranded DNA. And it was kind of a soft touch. It's not in any way a regulation. It isn't even tied to funding. It is a suggestion to the synthetic double-stranded DNA industry that they be on the lookout for sequences that could encode regulated pathogens. I'll mention that the reason this is not a regulatory framework is because the Federal Select Agent Program statutorily only regulates nucleic acids that could themselves give rise to select agents. So that would be full-length genomes of negative or positive sense single-stranded RNA. Those genomes themselves are regulated as select agents, but the subgenomic components of any select agent are not themselves regulated entities. So this, I think it wasn't entirely clear in 2010 what kind of impact this would have on the synthetic DNA industry. But in subsequent slides, I'll point out that I think that the guidance itself provided a good benchmark for the industry and has led to a wide uptake, thanks to our colleagues at IGSC. James, thanks for that. I'll mention before, Beth was overly kind and said that Matt Sharkey was very forward thinking in framing like the future perspectives of the guidance. I am just a scribe. I write down what my colleagues from the interagency come up with, and I try to aggregate it all into a policy document. So I don't want to take any credit for any of this. I wasn't involved in the 2010 guidance. That was Jessica Tucker, who led the process on behalf of ASPR. She's now a director of science policy over at NIH. So the 2010 guidance encourages providers to screen 200 base pair windows for regulated sequences. As I said, it's voluntary, and it throughout encourages providers to know who their customers are, and if they're providing their customers with a sequence that contains a 200 base pair window that is unique to a select agent or toxin or commerce control listed agent, then to do some follow-up and make sure that the customer is legitimate, has a legitimate use. And if malintent is suspected, it says, call the FBI. And my FBI colleagues have let me know that occasionally they're contacted, but it's only a few times, maybe a year. It's not every week that they get a call like this. So it looks as though there's not a huge problem with people with malintent trying to obtain synthetic nucleic acids. I'll talk later about the increasing availability of benchtop nucleic acid synthesizers and how we're trying to address that market. So thank you to the International Gene Synthesis Consortium, which self-reports that they encompass about 80% of commercial gene synthesis capacity worldwide. I don't think that they or we know that that's the case, but they're certainly a big organization. They have all of the big players in synthetic DNA, double-stranded DNA provision, and they require a harmonized screening protocol that is consistent with the recommendations in the 2010 guidance. So through the International Gene Synthesis Consortium, and without either U.S. regulations, international regulations, or a prescriptive funding mechanism, IGSC and others have managed to do a great job encouraging uptake of the guidance, both in the U.S. and internationally. I'll mention that although HHS and the federal government have not pursued a regulatory approach towards this guidance, states have. So California passed Assembly Bill 70 in 2021. It was vetoed by Governor Newsom, who I've read a summary of his remarks about this. I think his intention was that he didn't want California to have a regulatory framework that was out of step with the rest of the United States. I'll note that California has regulatory frameworks that are out of step with the rest of the United States on many other issues, including carcinogens and vehicle emissions. But this is an emerging field. I think the risk isn't seen as acute by political leaders. So I understand their reasoning. As a employee of the federal government, I have absolutely no position on whether Maryland or California should adopt such regulations. But I will mention, because I think it's relevant to the group, that that this has been investigated by states. There's a great overview of this from West and Gronval, published in 2020, talking about the bill in California. I'll also mention that Gigi Gronval spoke in favor of the Maryland bill in 2021 in front of the Maryland Assembly. Okay, so, and I'll mention also that those bills themselves pointed to either the IGSC harmonized protocol or to the HHS guidance. I think the Maryland bill was pointing to the HHS guidance, whereas the California bill pointed to the IGSC protocol. Once again, neither of these are law in either state at this point. So in 2017, Canadian researchers published an article indicating that they had purchased synthetic double-stranded DNA from a company, I think it was a Dutch company out of California. So it was GeneArt, which I think is Novartis, could be in Vitrogen. But in any case, they ordered synthetic double-stranded DNA that they used to chemically synthesize horsepox virus. This was a significant and unexpected advance, given that horsepox virus has more than 200,000 base pairs in its double-stranded DNA genome, and it has some structural elements similar to telomeres that were thought to be difficult to reproduce in the laboratory in order to give a transfectable full-length horsepox or orthopox genome that could generate horsepox virus. This, of course, being important because horsepox is 95% similar in sequence to variola virus, the virus that causes smallpox. And interestingly, for the application of the screening framework guidance, the 2010 screening framework guidance, although horsepox has many 200 base pair windows that are shared with smallpox, meaning that they're identical in sequence between the two viruses, since the guidance says that these 200 base pair windows must be unique to regulated agents, those windows are not unique to horsepox. Or to smallpox. They're of course also found in horsepox. So in follow-up, it turns out that everyone was doing their job. GeneArt understood that they flagged the order. They realized that these sequences were smallpox sequences. They understood the guidance well enough to know that since they're also horsepox sequences, there's no violation of the guidelines in that case. And they also knew that they were providing these sequences to legitimate researchers. And so they went ahead and provided them. The interagency at the time noticed this and thought the little kind of bullseye diagram on the right, we have a set of layers in how we want to affect best practices in this industry. We have regulations, which are the export controls in the federal solicitation program. We have the NIH guidelines, which are tied to a funding mechanism, and we have the sequence screening framework for providers. And we're thinking, what is the best way to address this? Nobody really wants to expand the select agent list to encompass a bunch of pathogens that are not essentially former BW program pathogens. But there are definitely risks associated with the provision of sequences that can contribute to pathogenicity or toxicity from unregulated pathogens. And so we decided to pursue revisions of the 2010 guidance that would encompass and mitigate the risks associated with this work and work like this. And just the impact, like the so what about the 2017 horsepox research is that it would allow, there's probably only a couple thousand people in the world who could carry out the work given what was published, but it allows some people, not just a couple of people, but it allows people around the world now to using synthetic double-stranded DNA to recreate potentially smallpox virus. Smallpox virus is the most regulated and access controlled material in the world. Of course, it only exists in 2 laboratories, one in Novosibirsk and one in Atlanta. And it's heavily regulated by the Biological Weapons Convention. So having this kind of out of access control puts everyone at risk. So that's why we sought to address this. Of course, there are other changes in the biotech landscape that we wanted to address and mitigate risks associated with in any revision to this guidance. The increasing ease of large-scale assembly, increasing ease of conversion between different types of oligonucleotides. Cost of synthesis is going down. There's way more companies producing these materials in 2022 than there were in 2010. The market is expanding rapidly. And then there are completely new risks, such as benchtop synthesizers and an emerging DIY community. And I'll say that as we considered all of these things, we tried to take as light a hand as possible, and I'll explain. We tried to spread the responsibility out throughout the entirety of entities that are involved in the provision of synthetic nucleic acids, in order to take some of the burden off of providers, and also in order to focus on building a culture of responsibility with what we are going to term sequences of concern. So we issued a couple Federal Register notices to get public feedback about whether and how we should modify the 2010 guidance. So in 2020, we issued a Federal Register notice that asked a lot of questions about, in these 5 or 6 different topical areas, what what could or should we do to change the current guidance? And then in 2022, we, after deliberating on the feedback for that first Federal Register notice for several years, we published a draft proposed guidance, and once again elicited public feedback on whether we were on the right track. We held a workshop in 2022. The EBRC helped us out with that. Got a lot of great feedback, and we've reached out to, as we have considered finalizing the draft and issuing as a new guidance, we've reached out to some people who are on the call here, including SIG Science and Aclid, in order to figure out if we're right-sized and doing the right thing here. So the links to those Federal Register notices are the short URLs on this slide. Okay, so these are the 7 topical areas that we requested feedback on in 2020. That kind of explains itself. So we wanted to know how we could modify the guidance in these areas to address emerging risks. And this is what these are the 5 broad areas where we modified the 2010 guidance and are getting ready to issue a final guidance document. It's in clearance at the White House currently. So what we expect will happen, and this talk is in no way meant to commit or to make evident the intent of the U.S. government, but what we think is going to happen is the White House will approve this draft or a modified version of it, and then it will come to HHS, where we'll put it through clearance, which essentially we send it up the chain to our assistant secretary and see if they like what they see, and then we'll issue it as HHS guidance. So this is kind of a sneak peek into what should be expected. This is very similar to the Federal Register notice that was published in 2022, but there are some modifications. I'll note that the scope has been expanded to provide recommendations for providers, for users, for manufacturers, for third-party vendors, institutions, principal users, and end users, by which I mean we are asking users to preemptively identify if they have, if their order contains sequences of concern. We're asking third-party vendors, principal users, and end users to verify the legitimacy of people that they transfer the materials that they've purchased from providers to, and to record those transfers. We're asking institutions to engage in some oversight of the use of benchtop nucleic acid synthesizers that are operated on their premises. And of course, we're asking manufacturers of benchtop nucleic acid synthesizers to start to verify the legitimacy of their customers. Not that we're saying they haven't been, but we didn't recommend it before. So we're just trying to be as proactive as possible in establishing best practices in this industry. So the definition of sequences of concern in the revised guidance will likely include all sequences that can confer pathogenicity or toxicity. This is potentially a problematic definition, which people smarter than me, such as Gene and the other bioinformaticists here, can speak to. And I'm going to have subsequent slides that talk about how we hope to mitigate the application of, or the potential challenges in the application of that definition. Of course, the 2010 guidance is limited to sequences that are unique to biological selectivation toxins or agents listed in the Commerce Control List. The types of oligonucleotides that are subject to the recommendations now include single and double-stranded DNA and RNA. We're suggesting the sequence screening window is decreased to 50 base pair, given the ease of the increasing ease of making larger constructs from small pieces of DNA. And then for we also suggest a screening window that's very small for batch orders, if those orders can be ligated and would have the appropriate overlaps for construction of larger sequences that are 50 base pairs or longer. So we also assume that that may be a little bit difficult for providers to implement. There are some record management and cybersecurity suggestions in the guidance as well that weren't present in the 2010 guidance. So I want to talk about some expected difficulties in implementing. So the sequence of concern definition, is inclusive of the 2010 definition, in that we kind of suggest that at a minimum, we suggest that you adhere to the 2010 definition. And then as soon as it's practical to do so, we recommend that sequences known to contribute to pathogenicity or toxicity, whether from regulated or unregulated agents, are treated as SOCs. So that's kind of a subset of the BSAT or CCL sequences. In the 2010 guidance, it was just suggested that anything from BSAT or CCL that is unique to those agents should be considered a sequence of concern. Whereas now we're saying, if you're going to go beyond those lists, the only sequences that you should consider to be of concern are those that contribute to pathogenicity or toxicity. I'm going to leave it to the bioinformaticists here to let you know how you might want to do that. And I just want to point out that it's further expanded in that we're talking about pathogenicity or toxicity that threatens public, agricultural, plant, animal, or plant products, or the environment. So that's a huge definition. I'll note that the government it doesn't seem that the US government is likely to host a unified screening database. We think that the US government will develop standards against which screening databases that are housed privately can be assessed for their suitability, something like an ISO standard, but not probably issued by the US government, not by ISO. But that is, of course, a major outstanding issue, is how could and should providers and users figure out what we mean by sequences of concern. We hoped to mitigate the implementation of this by asking users to preemptively let providers or benchtop devices know if their order contains sequences of concern. So they should preemptively say, yeah, I'm ordering a sequence of concern, and here's some information to verify my legitimacy. We think that users are best positioned to know if their sequences are sequences of concern. I mean, as a former laboratory scientist, I knew whether I was working with a viral envelope protein or a bacterial antibiotic resistance gene. That should be clear to any scientist who's placing these orders. We also are hoping to address the risks associated with benchtop synthesizers. So this guidance indicates that manufacturers should screen all customers purchasing benchtop nucleic acid synthesizer to validate customer legitimacy, and they should only provide those synthesizers to customers that have a legitimate need and have mechanisms in place to ensure that they are only opted by legitimate users. We're also suggesting that the devices contain either hardwired or cloud screening capabilities, and that they not be able to manufacture at least SOCs without that screening taking place. We have been meeting with the benchtop synthesis community to determine if that is possible. It turns out that all the benchtop synthesis manufacturers that we've met with already have a mechanism like this in place. In so much as they're trying to comply with the 2010 guidance. We understand that this mechanism becomes more complicated as they try to implement the revised definition of sequences of concern. Okay, so there are some questions here. We are also trying to establish an institutional responsibility to ensure that only legitimate users access the synthesizers and are able to manufacture sequences of concern. There are issues of how to deal with equipment sold prior to the guidance. One of the ways to deal with this is to verify legitimacy when purchasing reagents, especially reagents that are sole use for these benchtop synthesizers. And then we understand that some institutions have institutional security, biosecurity policies, or cybersecurity policies that prevent their devices from being able to access the internet. So we're suggesting that manufacturers come up with workarounds, and we've spoken with some manufacturers who already have workarounds for those issues in particular. So the last thing I'd like to point out, and it looks like I'm right on time ahead of it, that's great, is we included something in there that I think is pretty forward-looking. It didn't come from me. It came from my national security colleagues at FBI and DHS. And it's that, with the increased ability of machine learning algorithms to produce functionalities using entirely novel amino acid sequences, we expect that some actors with malintent may try to submit sequences that are completely unique, but which encode protein functionalities that are not themselves unique, like an analog of a bacterial toxin that has a completely novel sequence. And so we're suggesting that that the industry work to develop algorithms that can predict the structure and the function of sequences that aren't known otherwise. And just today, a fantastic group from Collaborative Pharmaceuticals, King's College London, and the Spetz Laboratory in Switzerland published an article. Oh, I gotta go forward a slide, sorry. There's ChatGPT for biology, what could go wrong? It's in the Bulletin of the Atomic Scientists, and it points to 2 articles published in 2022 that are deep language models for protein design, which themselves have published results that they are able to make functional proteins or regenerate the functions of natural proteins with extremely low identity in protein sequence to those proteins themselves. So I think that this kind of validates where we were thinking, that it's increasingly becoming possible to design new sequences that have a function that is already known, and we envision that people may, that actors with malintent may try to use these models to order bad functions or sequences of concern, functional sequences of concern that have unique sequences. And then we'll just point out, I would like to point out that some of the same individuals involved in that, including the folks from the Spetz Lab and King's College, published an article at the top here in Nature Machine Intelligence about the dual use of artificial intelligence for drug discovery, in which scientists at Collaborative Pharmaceuticals in North Carolina used or intentionally misused a toxicity predicting machine learning algorithm to generate a bunch of novel chemical weapons structures. So we think that the risk is very real, and we're pretty sure that the industry will act totally responsibly to mitigate these risks. And we are so grateful to have so many talented people working on this. That's all I got. If you guys want to learn more about ASPR, here are some links for you to look at. Feel free to email me. I'll say that I co-led the development of this revised guidance with Dr. Miriam Lekfishvili, who's also at ASPR. And Miriam and I would love to field any questions that anyone has about this. And here's my disclaimer once again, and I'll stop sharing the screen. So thanks a lot for your time today.